We'll be talking about some of the new species that have recently been added to the New York jurisdiction tracking list for IMAP. So just a few things before we get started. Today's webinar will be recorded. At the end of the webinar, you'll be receiving a link with a link to the recording, or you'll be receiving an email with a link to the recording, as well as any other links that we go over during the presentation. If you have any questions during the presentation, you can go ahead and put them into the chat box. If they're for any of today's presenters, you can address them directly, or at the very end, we'll have time for a little bit of Q&A on any of the species we go over today. So my name is Douglas Knopf. I am the Invasive Species Project Specialist with the New York Natural Heritage Program's iMapInvasives team. And I am joined today by a whole bunch of guest presenters as well. So we have Mitchell O'Neill, the invasive species biologist for the New York Natural Heritage Program. We have Liam Somers, entomologist with the New York State Department of Environmental Conservation, the Forest Health Research Lab. We have Dr. Patricia Kaishan, the curator of mycology with the New York State Museum, and Emily Timke-Benzinger, the aquatic invasive species coordinator with the New York State Water Resource Institute. If I got anybody's title wrong, just go ahead and correct me when you start your presentation. So today we're going to be going over a whole bunch of different species that have recently been added to the New York State iMapInvasives tracking list after a brief introduction of iMapInvasives and how we operate. And then again, at the end, we'll have time for some questions as well. So just to establish who we are, the New York Natural Heritage Program is the organization that is facilitated to be the data collectors and record keepers for all of the rare, threatened, and endangered species and ecosystem type information throughout New York State. And as one of the greatest threats to rare, threatened, and endangered species is the spread and establishment of invasive species, iMapInvasives and the Invasive Species Database Program was established to be the record keepers and data collectors for invasive species data throughout New York State. And the way we accomplish that is through the iMapInvasives database, collecting and holding all that information, and as well as our different data analyses and prioritization tools, things like the watercraft inspection program. And as invasive species is sort of this ever-evolving, complicated field of focus, we need to, occasionally make these updates to our species list. There's, new species that are being introduced all the time, things that are species that are on our radar in nearby states that might not be in New York State yet, or things that, have a high likelihood of being introduced sometime in the future. So to combat this, we are constantly doing research on other species that are already in New York that may not be qualified as an invasive species yet, and making constant updates to our species tracking list. So as of right now, iMapInvasives tracks 553 different invasive species in New York State specifically. And we'll go over a few of the ones that have just most recently been added within the past year. So we'll go ahead and get started with the red pine scale. Liam, if you're ready.
Yeah, can you hear me all right? Do I sound good?
Yep, we can hear you.
Excellent. All right. Thank you, Douglas. So yes, I am Liam Somers. I'm the entomologist for the DEC at the Forest Health Research Lab. And today I'll be talking about the resurgence of red pine scale in New York. Want me to just call out next? Okay, perfect. So red pine scale is native to Japan. It was first detected in North America in Easton, Connecticut in 1946. It was believed to have been brought in on infested nursery stock in 1939 during the New York's World Fair. And that infested nursery stock was brought back to Easton, Connecticut, planted, and then it began to spread through Connecticut, going into New York and Massachusetts. Here on the right side, we have a map from 1981 showing the distribution of red pine scale in New York. You can see there's pretty good coverage across Suffolk County, Nassau, and then up the Hudson Valley into Dutchess County. And I think a lot of that area that's infested on this map is likely red pine plantations, as it's just reaching up into the natural range of naturally growing red pine. But you, in this area that it's showing that's already infested, you really won't find any red pine unless it was planted in the last 10, 20 years. And even then, it's likely succumbed to red pine scale. You can click the next. So here's another historic map showing the natural distribution of red pine in New York State. You can see that there's pretty good coverage across the state, going up the Hudson Valley into the Adirondacks, reaching all the way to Clinton and Franklin County. And then there's a pretty good concentration in the Finger Lakes region as well. One of the things that I've noticed just doing field work over the past year or so, since I've been thinking about red pine scale, is how much red pine there actually is. So there's a chance that there's actually a lot more than that's being represented here. I mean, I know a lot of the red pine that I see, it looks to be planted, but you know, it's who's to say. It does seem to be in a lot more areas than you'd really think. Here's an interesting quote from 1977 in a New York Times article featuring Fred L. Gertrude Jr., who was a forester for New York State DEC. He said, "We have no way of controlling it. The real problem with this insect is that if it continues to spread, it will one day kill every red pine tree in the North American continent." So they're pretty pessimistic about it in the 70s and 80s, and it's pretty strange that it's kind of gone unheard of for that long. And that was really up until this past summer. But we were thinking it was probably hitting that temperature threshold that a lot of insects run into, where they won't expand northward anymore because it's hitting a certain minimum temperature that's killing them off. For red pine scale, that's about -10 degrees Fahrenheit, and that's generally what we were thinking was keeping them south of Dutchess County up until this past summer. And that is when we were contacted by a homeowner on Lake George, saying that the island across from his property was, it had a bunch of red pine trees that were flagging. And when we went there to see what was going on, we noticed that the site wasn't that great, so I'm sure that did have a lot to do with it. It was pretty, you know, exposed soil, very compact. All the roots were exposed as well and just nicked up from people like camping and walking around on the island. We were thinking that was the main cause of the decline in these trees. We did see a little tuft of white wool at the ends of one of the branches. You can see myself actually holding the branch in the picture on the left. We clipped that, we brought it back to the lab, and there's a little video here if you don't mind clicking play. It should just be at the bottom there. Yes, so once we brought that back, we found these little ladies, which are adult female red pine scale, and they were just crawling all around that branch, primarily in and around the wool that they produce. So about a month later, we went about 10 miles west of that initial detection to a fairly substantial red pine plantation at Finger Lakes Wilderness area. And upon arriving, we immediately noticed all the red pine there was either dead or dying. And we were able to find a couple of branch samples that were just knocked off in wind or something like that on the ground. We saw that characteristic white wool, and we were able to extract some specimens. They were dead, but we were able to extract some pretty dehydrated specimens from there that we sent to Cornell, and it came back that both that initial detection on Lake George and this one at Finger Lakes was a match to red pine scale. So I should say match in a loose term because this is actually pretty interesting. So it came back as a 99.91% match to red pine scale of the genetic banked sequence for red pine scale, at least. you might think that sounds very similar, which obviously it is very similar. And we're thinking maybe that its DNA diverged from that original sequence from its native area over the past 80 years since it's been here. It could even be its own species. We're not really sure. So for right now, we're characterizing it as the red pine scale complex until we're able to learn more about it, which we're hoping to fill in some of these knowledge gaps as we continue to research and collect specimens and run some of that DNA with our partners at Cornell. Next. So I won't get too deep into the life cycle, but I guess the main takeaway is that there's 2 generations a year. There's a growing season generation and an overwintering generation. The main point is to look for it in August through September. That's when it's most visible. That's when the adult females are making these the fresh ovisacs, which are a lot more clear to see. Because as time goes on and they start to get beat down a little bit, they're just a lot more difficult to detect. But when they're fresh, they're pretty clear. They look similar to that picture I have up in the center there. It's not a great photo. My cell phone's pretty old that I took that with, so I should get a better photo than that. But generally, it's easy to see, I guess, in these months of the year in an infested tree, and that's generally around the ends of the branches is where you'll find this wool, and it's like the base of the needles area. Sometimes you'll have to fleck off some of the bark scales because it can be like tucked in there a little bit. They can be kind of tucked into the red pine cones as well. You'll just kind of have to look around, but the main thing to look for is that flagging branch, and it's usually sporadic throughout the tree. There's no real like pattern to it. It's just kind of here and there wherever they land and start to feed on. That'll cause that flagging, and then you should see this wool start to appear. So our main goal with red pine scale is to protect our natural red pine stands. So right now this summer we're doing some red pine stand health assessments and some plot work to determine the distribution and damage severity. It's just something that we haven't really documented before. A couple years ago, we did have some of my colleagues go out there and do some of this work, but they didn't find it anywhere. They also weren't necessarily looking in the right spot. To my understanding, they were kind of looking all around the tree rather than the ends of the branches there, where we found it to primarily be. But outside of just like the mapping and figuring out the damage severity, we're potentially going to be managing some of these infested plantations that are harboring these large populations. So in the 1930s, the Civilian Conservation Corps planted a bunch of red pine plantations with the idea of using that for telephone poles, electrical poles, that have just kind of since been forgotten about since World War II started, and they're all pulled away from there. We have all these red pine plantations across the state, and you got to think like a pretty dense monoculture forest stand. If you introduce an invasive species to that stand, they're just kind of run wild throughout there and build up a large population and bleed over into the natural areas. So that is something that we want to we want to try to mitigate as best as we can. And lastly, of course, iMapInvasives, our favorite tool, just add a red pine scale to the list. It makes it so much easier for us if reports are made through there. But if you see something suspicious that you think might look like red pine scale or any weird things going on with red pine in general, feel free to reach out to the research lab with our phone number, our email, send photos, videos. The more information, the better. You could reach out to me directly as well, whichever way would be much appreciated. That's it for me.
Great, thanks, Liam. Again, if anybody has any questions about any of the species we go over, you can go ahead and put those in the chat box. Liam, if there's any questions that you can answer in the meantime before we get to the end, feel free to do so, or you can wait.
Will do.
Right. And then up next, we have Dr. Patricia Keshin with the golden oyster mushroom.
Hello, thank you for having me. Hi everyone, I am the New York State mycologist, or the curator of mycology at the New York State Museum in Albany, and I've been in this position since summer of 2023, so still feeling kind of new to it. And I'm here to talk to you about a pretty interesting situation, the golden oyster mushroom, Pleurotus citrinopiliatus. And this is an unusual situation in mycology because there are no other currently known escaped mushroom cultivars that have become invasive in North America. There are many examples in history of fungi that have been introduced and run rampant. Obviously, the chestnut blight is one of the more well-known, but the fungal pathogens that have been introduced and become invasive are all microfungi. And this is an example of a fruiting, like the mushroom in the true sense of the word, and a macro fungus that has been also not accidentally brought to North America, but intentionally cultivated. So there are several layers of intrigue here, but it is also a pretty emerging issue that has popped up, no pun intended, in the last decade or so, and seemingly has really accelerated in the last few years. So I'm just going to give just a basic primer on this mushroom, but there isn't a ton of concrete data about the situation, and it's very much like emerging, and the mushroom community is just starting to grapple with it. So their common name is golden oyster, or Pleurotus citrinopiliatus. Citrino meaning lemon, yellow, and pileatus is the cap. So it's a yellow mushroom, and you can go to the next slide. I'll just talk you through a little bit how to identify it, because it is actually not terribly tricky to identify as a macro mushroom in North America. It is golden, as the name implies, and they grow in large gregarious clusters. They're typically found on hardwood. The caps can be pretty large. They can be several inches across, and they have these cream to slightly pale yellow gills on the underside that are decurrent, meaning they run along under the cap and then down partially the length of the stem. They can have pretty central stems that can be central to relative to the cap, but they also can be eccentric, coming off to the side and growing in an almost shelf-like fruiting body from the substrate. But there is some variation in the exact sort of growth form of the fungus. But they're pretty conspicuous. I often these days see them from a great distance. Like, I can be driving my car on the down the road and see them out of the corner of my eye on a fallen log or a standing dead tree, in a scrap of forest, like in the roadsides and stuff like this. So they're pretty easy to see. They're very bright and conspicuous. Even when they've passed their prime and are starting to decay, they still are pretty golden. And there could be hundreds of them on a single tree. So they're saprotrophs, meaning they decompose dead or dying organic matter. So they take advantage of a tree that's already dead or is close to death, and they're a white rot fungus, so they're breaking down the lignin in the tree, and they're just incredibly gregarious. And abundant. Okay, next slide. Just another angle of them growing. These are all photos I've taken myself in the Hudson Valley, variously located. And they're typically on oak, ash, and elm, and other hardwoods. But it is like an emerging, again, there's a lot of anecdotal information, and we're still trying to collect as much data as possible. And I'll get into some of how we can, get support collecting some of that data in a minute. But I'm typically seeing them on those 3 types of trees, especially seemingly oak seems to be the most common. Well, partly because the tree is more common than the others in my area. So yeah, so it's something that can easily be identified, and conveniently they're also edible. So which is part of the problem, is part of how they've become introduced. So they're attractive to a lot of people as cultivars because of the reason, because of their really quick growth, their generalist sort of capacity to break down substrate, and the fact that they can just grow really, really aggressively and really quickly in a lot of, and so I'm finding them also, like, they're not very seasonally affected. So I can find them I've seen them already this spring, and I find them deep into the fall and throughout the whole summer. So they're kind of using at least like 6 months of the year in New York to be growing. Whereas, as most of you probably know, a lot of other mushrooms have much more narrow growing seasonal windows. So that also makes them attractive for cultivation. We're actually okay, you can go to the next slide. So just another angle, just wanted to show you a bunch of angles so you can get to know them, and go to the next one. So here's a picture of them growing on a substrate. So we believe that they were introduced to the wild through cultivation, and there's a correlation between their emergence as established invasives and the sort of surge in popularity of mushroom cultivation, both at a commercial scale and I think really critically for this species in particular, the boom in at-home cultivation. So there are a lot of small businesses that focus on growing mushrooms that also sell grow kits for this species in particular, because it's easy to grow. And so we actually think that one of the primary means by which this fungus is being introduced to the local ecosystems is by at-home cultivation, by just anybody who goes to the farmer's market and picks up a little block. So these mushrooms, you can see not super clearly, but they're growing on these blocks that are like compressed hay and sawdust and things like this, and then they're inoculated, and then they can kind of grow repeated flushes of mushrooms until the mushroom has exhausted its substrate. And it's, if for not being an invasive species, it's a very fun thing to do. It's billed as an environmentally friendly thing to do. People are instructed to simply discard the spent grains after they've run, the mushrooms have exhausted them, and it's kind of pitched as something that can enrich your garden and all these things, which is true. But unfortunately, what we're seeing is some really significant adverse effects of this. Okay, next slide. Just some summary information that I've just said. So this is a fungus that is native mostly to North and West Asia, so parts of Russia and Western Asia, Western China, and it's kind of commonly cultivated in those regions as well. And then in the last decade or so, you've seen an increase in cultivation here in North America. Anecdotally, I grew up in Westchester and Putnam counties. Then I went to graduate school in Syracuse from 2015 to 2020 at ESF. And I never saw golden oysters until 2021 in the wild. So I have spent a tremendous amount of time looking for mushrooms throughout New York, and it's only been in the last 5 years that I've started to see these fungi. And now I spent 2 years as a professor at Bard College, which is in the mid-Hudson Valley. And this is anecdotal, but I would say that there were times, weeks out of the year, 2 years that I was there during like the warmer weather, that this fungus was the most abundant visually detectable biomass. So, anecdotal, but if I walked in the forest, the most biomass I was visually seeing was this fungus, which is really alarming because it went from, in a very short amount of time, from I've never seen this fungus and I've spent my whole life combing the forests looking for fungus. And now suddenly it's like you can barely walk through certain forests without seeing it. Another anecdotal piece, like a bit of information, is that it does seem to do well in slightly disturbed or very disturbed habitats. So for example, I do conduct a regular weekly inventory of fungal diversity in a forest near the, like in New York, near the Vermont border. And by no means is it a pristine forest, but I've never seen this fungus there. It's like a pretty decent, a pretty biodiverse place with a lot of different tree stands, and I would say a good amount of fungal biodiversity. But I will see this fungus in the strip of forest between Lincoln Park in Albany and the New York State Museum, which is, right in downtown Albany. So I'm seeing this fungus mostly in pretty disturbed, like roadsides but also in deep into the forest, but in forests that have like really heavy legacy usage. So we are concerned because we have some evidence that this fungus can really rapidly decompose the substrate, and that pace of decomposition will be outcompeting possibly the native decomposers. So other saprotrophic fungi, macrofungi, but also microfungi and other organisms, entirely other kingdoms that are responsible for decomposition. We are concerned that this fungus can sort of just plow through the decomposing wood, gobbling up vital nutrients and exhausting the substrate without the ability of without allowing other organisms to establish themselves in the wood concurrently or even maybe afterwards. So there's a real concern for this. So actually go to the next slide, please. One of the first people to do a lot of research on this fungus and its an invasive potential or status, it was Andy Bruce, and she did her master's thesis in 2017, and I've linked to their thesis here, and they have all sorts of data related to the genetics of this fungus and some of their survey work. And I took this text from their blog. They write about this situation. So in their thesis, they looked at whether this was a single clonal lineage of fungus. As some of you may know, like fungal reproduction can get really complicated. Sometimes fungi are just reproducing asexually. Sometimes they're sexually recombining, and they've kind of all sorts of combinations, within that. But in this case, it seems as if, based on Andy's research, that they were sexually reproducing and therefore obviously interbreeding and having sexual recombination and creating non-clonal offspring. So they found that there was actually some genetic variability across the commercially cultivated strains and the strains that they were finding out in the wild. But there were particular strains that seemed to be much more represented in the wild specimens that they encountered. So there were seemingly a few lineages of cultivation strains that were successful at escaping and establishing themselves. And then her research also noted that there seems to probably be multiple repeated events of introduction, because they were finding strains that were at really, really different, distant locations, and in some cases almost 1,000 miles apart, where the same strain, really similar wild isolates were found at having been seemingly maybe they were cultivated, and then introduced separately from separate cultivation production, I guess events. So this is concerning because, we think that there's just repeated, this is constantly being introduced because of all of the home and small scale growing operations that are literally encouraging people to do this. There were so they mentioned some rumors of possible explanations of how this could have been, if there was one singular event that led to the introduction. And that some of their research sort of dispelled those rumors. We think it's repeated, smaller introduction events. Okay, next slide. So what can you do about it? There's another scientist who is studying so Andy Bruce studied at the University of Wisconsin La Crosse, and there's also a University of Wisconsin-Madison lab run by Dr. Ann Pringle, and she has a graduate student who is currently focusing her PhD research on the invasive nature of this fungus. So she put out a call for how to support her research so we can get more data to like fully understand this picture. And I will just say, I meant to say this in the beginning, in my current capacity, I'm not officially researching this. I don't have a current project myself, so I'm directing you to this graduate student's project. This lab has been doing work on the invasive capacity of this fungus now for a few years, so they're well positioned. And so maybe what we want to do is use iNaturalist to be tracking wild encounters with this fungus. So not specifically cultivated stuff. So if you're walking in the forest and you see it, they have an iNaturalist project that they want you to be using. And they're also asking for fungi. Specifically, New York is a state that they've listed that they specifically want specimens from, and they just are hoping that you can collect them in a bag, refrigerate them, and then take some notes as to where you found this geographically, and if you know the host tree species. And then send the specimen to the Pringle lab in Wisconsin. So I have their email there if you have any questions about that, and the address also of the lab. Just again, this is sort of just like we are as a mushroom community trying to figure out best practices. Some of us have discussed the fact that when you collect a mushroom, if it is mature, it is constantly sprinkling its like millions of spores around. And so if you pick it and then walk with it, you might be accidentally just helping it proliferate. So it's a really tricky problem. Some people have proposed like kind of wrapping a plastic like a little like plastic grocery bag around them and sealing it and tying it before moving it. And I think that kind of makes some sense to me. Like in general, actually, we often teach people about how when you pick mushrooms, you're not really killing it. You're actually, if it's mature, you're giving it a chance for its spores to disperse. But in this case, that obviously might not be a good thing. So we're also doing some outreach with mushroom growers to try to stop growing this and to certainly especially stop selling home cultivation kits. So the outdoor growing in particular, just seeding just every crop, billions of spores, it's a little bit overwhelming to think about. So yeah, that's what I have for today. I'm also happy to take any emails. I can put my email in the chat because I realized I forgot to put it in the slide. But thank you so much, and let me know if you have any questions.
Great, thanks, Patty. A lot of great anecdotal information there,
Mhm.
and we'll be interested to see where the research goes with that. So up next, we have featherfoil or Houtonia species, presented by Emily Timke-Benzinger. Emily?
Hello, can you guys hear me?
Yes, we can hear you.
Oh, perfect. I'm just going to keep my camera off right now. I have a pretty spotty internet connection, so I'm worried that'll send it over the edge, and I won't be able to connect anymore. So yeah, I am Emily Timke-Benzinger. I am the Aquatic Invasive Species Coordinator for New York State CEC Region 7, and that is with a partnership with the Cornell Water Resources Institute. So Region 7, we're kind of the Eastern Finger Lakes area. And so if you want to go ahead to the next slide. So the species that we're looking at, Houtonia, it was first brought to our attention by a report on iNaturalist, and in the Corning, New York area. So myself and a couple of my, summer crew, we went out there, we took some photos, collected a sample. Actually, if you want to just click the next photo, should pop up. So this is a photo that was uploaded to iNaturalist. Clearly, it's something that we had not seen before, so that raised some alarm. We went out to collect a specimen, and we sent that in to get some DNA testing, and it did come back positive as Hatonia palustris. So this species, it does have a, I guess, a native counterpart or kind of a native lookalike. So American featherfoil, and that's the native species found in the U.S. And I do have in a couple slides just a quick, identification way to tell the two apart. So a couple other names for Hatonia palustris is water violet or featherfoil. I think most commonly I've heard people use water violet to talk about it. So this is native to Europe and Northern Asia. It's actually considered endangered in some parts of Europe. And it is popular in the aquarium trade in the US and Canada. So that is how I am assuming, we don't know for sure, that is how it got to Corning, New York. So we went out to this site, and basically it was just in a tiny little pond, almost like a koi pond that someone had in their front yard. The whole pond itself was maybe 5 feet by 5 feet, no deeper than maybe 2 feet deep, and it was completely covered in this species. So if you go to the next slide, or not next slide, next photo, that is pretty much what we saw across the entire pond. So like I said, we went out, we collected a sample. There were no flowers present when we went out, because we didn't get out there until maybe mid-August or so, and this plant typically flowers in May into June. So we kind of missed that. And then this plant was also, by the time we went out there, it also was kind of emergent. So it was growing in the water as a submerged plant, and then it also was growing out of the water in some muddy shore area just around the small pond. And one of the first locations that this plant was found was in New Hampshire in 2021. It was just found in a lake in a couple of bays, and then since then it's also been found in Maine, Michigan, and Connecticut. So somewhat, spread around the Northeast. And then again, we had that positive identification for the first time in New York State in 2024 in Corning, New York. So just a couple quick notes about how to identify it. So most of the plant is submerged. It will become emergent, later in the season and as it flowers. Whorls of submerged feather-like leaves that spread under the surface, and they can grow to be about 3 feet long. This invasive species produces whorls of submerged foliage during the spring months in somewhat dry conditions, and then in the summer it will produce spikes of white, pink, or pale purple flowers. So that was that previous picture that you saw that was from the homeowner. In the water violets, they can grow as tall as about 20 inches above the water surface, so that flowering spike can grow pretty tall. And again, it can also grow, outside of water in, moist, muddy areas. And like I mentioned before, we didn't have any inflorescences present when we went to go and ID the species ourselves. So we did have to collect that DNA sample. So if you just look at it, it doesn't look super, the best way to ID it is with those flowers on it. So if you want to go to the next slide. Again, that's just another photo of what we were seeing. And so the emergent leaves that we were seeing, they were a little bit thicker and flatter and wider. But again, this was later in the season. It had been pretty dry as well. So we're really only seeing those emergent leaves. We didn't really see anything super fine. But if you look at the photo from the landowner, you can see that those leaves were nice and fine and kind of matched better to the description that has been you know, that's a little more widely available if you try to ID the species, like either online or using some ID books. So you can go to the next one. So here is on the left-hand side, that is the some plant specimens that we collected, and then on the right-hand side, those are photos that were, or some plants that were collected from New Hampshire. So our ruler is a 12-inch ruler. The one from New Hampshire on the right-hand side, that's a 15-centimeter ruler. And so again, we did collect at slightly different times of the year. So I think that that kind of has something to do with why the leaf structures are so different, or why they look a little bit different. And so I would like to go back and try and collect something within the next, you know, within this next month, to try and actually get a press or get a flower sample that we can press and keep for ourselves for future identification, and then also, you know, send to our state botanist as well. You can go to the next one. Okay. So these are just some of the lookalikes. So the native species, which is the American featherfoil, Huttonia enslida, which is on the right-hand side, you can see it does have these inflated inflorescence axes, whereas the invasive one, it does not. The flowers on the native species are also much, much smaller. You can't really see them super well, unlike the invasive species, which has some, you know, larger, more showy flowers. And then a couple other things that I think it could definitely get mixed up with would be some milfoil, so the Myrophyllum family. So especially parrot feather, because this species can also grow in the submerged area and then also up on land as well, much like a parrot feather. And then also some other invasive milfoils, especially when it's just submerged and you see those feather-like leaves in the water. And so I believe that the Huttonia palustris is going to be added to the next edition, or if it hasn't already, be added to the next edition of one of the, you know, larger ID books, the Hellquist book. And then I'm just trying. Okay, and then also, as far as management for what we're planning to do in this particular site, that we did find the water violet. So it is a super small area. We've spoken to the landowner who is, you know, who did actually upload the photo onto iNaturalist. They've been super cooperative with us and, you know, willing to work with us. They let us come out, take samples, etc. We spoke with them. We, you know, told them not to move the species, not to give it to their friends or anything like that. You know, that it is technically an invasive species. So at this point, we don't have any plans to manage it. However, that might change. And as far as I know, we have not had any other detections of the species in New York State. But we also, you know, we're keeping an eye out for it to see if we do see anything. And then the next slide, or the last slide, that is just some resources. So New Hampshire, which is one of the first states to find and identify this, they have a couple of fact sheets out there. And then there is also some folks at U.S. Army Corps of Engineers have been working on determining, you know, if this species is responsive to any type of aquatic herbicide that could be used for any large-scale infestations. And I think that that was everything I had for this. And I realized I also did not put my email on the slide, so I can put that in the chat. But if anyone has any other questions, please let me know.
Great, thank you, Emily. And I hope you have good luck with collecting your samples later. And I believe that brings us to our last presentation section, which is going to be on Hamilton's Spindle Tree by Mitchell O'Neill.
Yeah, thanks. And I actually wanted to make one quick comment on the Huttonia first. So thank you, Emily, for mentioning the native Huttonia inflata. I just wanted to mention that if you happen to find that, we'd still be interested in that in the New York Natural Heritage Program. So not for iMapInvasives, but for the rare species database. So Huttonia inflata is considered a rare species in New York State. It's mostly like downstate area in Long Island. So you're not super likely to find it in most of the state, but if you do, I'll put a link in the chat real quick. And it's an S2 species, which means it's imperiled, and it's a state threatened species. So also a word of caution to make sure you're really sure. If you think it's Huttonia palestris, make sure you're really sure before considering management. And so yeah, so now I'll talk about Euonymus hamiltonianus, also called Hamilton's spindle. And so some of you are probably familiar with several of the Euonymus species that we have in New York State. This one, it could have been here for a while, but it's kind of recently become noticed by botanists in the region. So those are just a few pictures of it to give you an idea. It's this woody small tree or shrub. So next slide. And I want to mention that this was one of the species highlighted by the New York Botanical Gardens Emerging Invasive Species Alerts. So they're working on a new guide for a new flora for the Northeast. And in looking at herbarium records and surveying plants in the field and stuff, they've identified these 5 plant species, which seem to be relatively new to the region, but are spreading somewhat rapidly, but they're not completely widespread yet, or at least the data is not showing that at this point. So they highlighted these 5 plant species, and we added those all to the IMAP tracked list. I won't talk about all of them today, so I encourage you to Google those. But one of them was this Euonymus hamiltonianus, or Hamilton spindle tree, which is in the bittersweet family. Next slide. And some context, it's native to Asia, and it's been introduced as an ornamental. It's very pretty, in my opinion. And there are anecdotes of it spreading, and in places where it's found, it tends to be abundant. But we don't really know impacts yet. We don't have much information. We're just kind of being cautious and assuming that it could be invasive, simply because there are several other species of Euonymus, of non-native Euonymus, that have had invasive impacts. Next slide. And one important thing to talk about is there are all these other Euonymus species, and some of them look very similar. It's very possible that there are misidentifications, and this Hamiltonianus species could be underreported or misreported as other species, so it might be more common than we think it is. But a lot of you may be familiar with the Euonymus alatus burning bush with the bright red leaves and the corky wings on the branches. And there's also Euonymus europaeus, European spindle tree, which looks even more similar to Hamiltonianus. And then there are other non-native species as well. The Japanese spindle tree isn't as widely naturalized, and the winter creeper is a vine, so they're like not as likely to be misidentified, so I won't talk about them. And then I also want to mention that we do have a couple native Euonymus species. So the 2 on the right, those are kind of different looking. They have like bumpy, spiky looking fruits. They're very cool plants, and they're not common in most of the state, and they look a little bit different, so I'm not going to talk about those. But one species, Euonymus atropurpureus, or the wahoo, looks very similar to Euonymus hamiltonianus. So I'm going to go through some features of Hamilton spindle tree, and I'll also be mentioning the things that help you distinguish it from the similar looking Euonymus trees. Next slide. So first I'm going to talk about the bark and branches and just like the overall form. It's kind of, you can see in that picture, it's a single trunk, small tree type form. It can be more shrubby as well. And the features I'm going to talk about in terms of bark and branches, they aren't necessarily great diagnostics for which euonymus it is. So you'll see some of these traits in the other euonymus species, but it's a good place to start because they're kind of some of the distinctive features of euonymus. So next slide. Yeah, thanks. So you can see the bark has that kind of stripy pattern and the new growth is very green. Next. And this euonymus won't have the big corky wings that burning bush has, Euonymus alatus, but it can have very small, subtle ones. They kind of just look like corky stripes. So you can see that very fine green stripe that the arrow is pointing to. So those are all clues that this is a euonymus. Next slide. And again, those won't help you tell which euonymus it is. So for that, you look at the leaves, the flowers, and the fruits. So starting with the leaves, they are oppositely arranged. You can see those leaves are occurring opposite to one another. Those individual leaves, they are a little bit bigger than what you would see on a euonymus alatus, if you're familiar with that, but somewhat similar in shape to other euonymus species. And there's kind of some patterning on the leaves on that picture on the left. That's just from an insect. That's not a real trait of this species. They're just kind of plain green leaves. And in that close-up picture, this observer has folded, they're showing the underside of the leaf and they've folded it. So that's a really good way to see whether there are hairs on the underside of the leaf. And that's important because that's one way you can tell between this and Euonymus atropurpureus, one of the native species. So if it has hairs on the underside, it's the native one. If it's hairless, except maybe some hairs on the veins, but if it's mostly hairless, then it could be Hamiltonianus. Next. And next. And so just some other traits that might help as you get into the fall, the leaves do turn reddish, orangish, kind of brownish, but they don't get that really bright red color that you see on Euonymus alatus, the invasive burning bush, or the native Euonymus atropurpureus, the wahoo. So the leaves are important, and then next the flowers. So the flowers are pretty inconspicuous. They emerge right around now, like, late May, June. So they're these tiny white flowers. And if next for a close-up, but if you look at them closely, they have 4 white petals. And if you look really closely, you can see the anthers sticking out of the flower, and they are sort of reddish or purplish, very tiny. And that's a good diagnostic feature as well. So next. So you probably can't see it in those little pictures, but those are some of the lookalikes. The two of the other invasive euonymus species have pale colored anthers. So they're not those little purple reddish balls like on the left, they're white or yellow. And then underneath the native wahoo, atropurpureus, is much easier to tell because the flowers are very vivid, dark red or purple. Okay, so next. And then another thing to look at is the fruits. So the fruits are very showy. This, I think, is the most beautiful part of its yearly cycle. So like other euonymus and other species in the bittersweet family, there are these capsules that kind of mature and break open, and then there's those 4 arils inside. So they're, seeds covered in flesh. And on Euonymus hamiltonianus, they are a very pale pink capsule and then bright red arils, which are the little berry things inside. Next. And so you can distinguish it from other euonymuses at this stage. So for the European spindle tree, those have pink capsules as well, but the arils are bright orange. For Euonymus alatus, The arrows are also orange. They're very orange in that picture. They can be a little bit more reddish, so sometimes it's harder to tell. But the capsules look pretty different. So they're kind of more collapsed and darker colored. They're not the kind of puffed up bright pink ones. And then for Atropurpureus, I think it's very hard to tell the fruits apart for these two. So in theory, they all start pale and get darker. And in theory, the Atropurpureus capsules, they get to a darker shade of pink. But in my opinion, like I can't really tell. So looking at pictures, it just kind of depends on the lighting. They all kind of look the same. So in this case, if you're using the fruits, which, and I should have mentioned, these are all pictures of the mature fruits. They all kind of start paler and then mature into their pinker colors, and then they break open and you can see the seeds. It's not really until they get mature and break open that you can really use them to tell the species apart. If you're looking at, and that happens in the fall, and so if you're looking at the fruits, you also want to get a picture of the underside of the leaf. So that will help you, if it's the pink capsule and the red arrows, you won't know if it's Atropurpureus or Hamiltonianus, but if you could look at the underside of the leaf, then you'll be able to tell because you'll see whether or not there are hairs on the bottom. Next. And just to review it all real quick, so it's a small tree or shrub. New growth is green. The leaves are opposite and mostly hairless on the underside. In the spring, they have small pale flowers with purple anthers. And in the fall, they have those light pink capsules with red arrows. And the leaves turn reddish brown, but not bright red. So in terms of what to photograph, general pictures of the leaves and branches and bark can help you tell like that it's a euonymus, which is good. But to actually tell which species, you'll always want a photo of the underside of the leaf folded over, like on the right side. And then if you have fruits or flowers, take pictures of those as well. And for other resources, you can look at those, and New York Botanical Garden Emerging Alerts or the Michigan Flora has a good key on that. Next. And so we did a highlight of several of the species added today because we wanted to be able to do a deep dive into some of them, but there are a lot of others. So I just wanted to put them up on the slide. I don't have time to talk about any of them. So if you want to take a picture or look for this when the recording comes out and learn more about these species, feel free to do so. Yeah, I won't mention anything more on that. And so I think that's my presentation, and so we'll pass it back to Doug.
Great, thank you, Mitch. And also thank you to Liam, Patricia, and Emily for your presentations as well. A lot of really great information on some of the new emerging species for New York State. For the last section of the we're going to be changing pace just a little bit, sort of in the theme of identifying characteristics for reporting species to iMapInvasives. I wanted to go into a little bit of information about one of the features that you can utilize while you're surveying for certain species out in the field this season. So there's a lot of invasive species that have one specific species and that's it, but there's also many species where there's multiple species within the same genus. So an example would be something like knotweed, where you have 3 different species of knotweed, being Japanese, Bohemian, and giant knotweed. That can look very similar a lot of times of the year. Something like knotweed, you need for it to be in flower or to get very close-up detailed pictures of some very minute identifying characteristics in order to be able to tell the species apart. So for the ease of reporting some of these species into iMapInvasives, we've grouped a lot of these these genuses together so that you can report them as just the genus without specifying which species it is. So if it's a time of year where you don't have those identifying characteristics, you're still able to report the species and have it be a confirmable record in iMapInvasives database. I won't go into too much detail about the specific things for each of the species. For more information on that, you can look on the iMapInvasives website or look into your local PRISM website for more specific species ID information. Again, just a few examples of species that have this trait. things like knotweed, where you have the 3 different species that look very similar most times of the year, except when they are flowering. So you have the ability to just report it as knotweed species unknown in the iMapInvasives database. And again, this makes it so it can be confirmed and show up as a confirmed record rather than being unconfirmable as a specific species. And then the honeysuckles as well. There's 8 different species of honeysuckle in New York State, 7 of which are bush honeysuckle that all look very similarly most times of the year. And for that one, you can just report as honeysuckle species unknown or Lonicera sp. These are the other genuses that we have in iMapInvasives that have the ability to be reported as just the genus. So swallowworts, silvergrass, wisteria, bamboo, jumping worms, knapweed, privet, oleaster, and land snails. Land snails being one of the newest species to be added to the IMAP network that was on Mitch's last slide that he was talking about. So for each of these genuses, if you don't have the right timing, if it's not in flower, or it's past the point where you can get those really key identifying features that would let you identify the species, you can just look for some of the more general identifying characteristics that you'd be able to tell the genus from when reporting to IMAP. This is a good thing to teach like volunteers or field crews that are going to be conducting these surveys throughout a large period of time and might miss that window where these species have their identifying characteristics. And with that, that brings us to the end of today's webinar. So I just want to say thank you again.